All right, today we're going to talk about mitosis, which is also known as cell division. The picture that you're looking at right now has, I believe, onion cells or pictures of onion cells. Later this year, we may actually get to do a little lab where you guys get to see this stuff yourself. But the reason that people often look at onion cells is because this is a place, or actually in the roots of onions, this is a place where we see lots of mitosis happening a lot. So you'll see mitosis anytime that you have lots and lots of growth. When the roots of trees or the leaves of trees are starting to grow, especially in the spring, you've got all sorts of mitosis going on. So basically, mitosis is just the turning of one cell into two cells. And the way that they do that is we divide up the genetic material. This is a little bit different from something we're going to learn about in a second, which is cytokinesis. Cytokinesis is where we divide all the other stuff up. Mitosis is just dividing up the genetic material. The cell has to grow, and it has to make two times of everything, and that includes the DNA. DNA, again, is what we use to transfer our hereditary information. Mitosis is relatively quick. It actually only takes about 10% of the cell's life by some estimates. Most of the cell's life is actually spent in just some different phases of growth where we're replicating the DNA and we're replicating all of the different organelles that are inside of the cell, so that later on during mitosis we can divide those up. There are four main phases of mitosis. The first one is prophase, and then metaphase, then anaphase, and then telophase. All these little phases of mitosis, though, are just a tiny part of the cell's life cycle. Beforehand, we have something called interphase. An interphase is where we're growing and we're just preparing for the division process. Right after mitosis, when we've finished telophase, we go into something called cytokinesis. And cyto, again, means cell. This is basically where we actually do the cell division stuff. This is where we take extra organelles and we move them from one side of the cell to the other so that we get two completely identical cells. So the first phase, again, is prophase, and basically in prophase, what you're looking at in the little diagram on the top is the nuclear membrane, which is the little area that kind of surrounds the nucleus. It keeps everything in the nucleus, including all the DNA. The nuclear membrane is breaking down. At this point, you guys can kind of see that those little X shapes, the blues and the reds, those are actually our chromosomes. So at this point, when we've started prophase, the DNA has actually condensed, and the picture below is some kind of a plant cell. You can kind of see that everything has condensed in there, and it's all in that little circle. So this is a real-life picture of prophase, if you want to see it. After everything has condensed, we've got all of our chromosomes there, the nuclear membrane totally breaks down, and we actually go into metaphase. Meta means middle. So when you think meta, okay, metaphase is where everything lines up in the middle. All of the chromosomes are lining up in the middle of the cell. Another thing that you guys can see there on that top diagram especially, you can kind of see those little lines, the little fibers that extend from those little spaghetti noodle looking things. Those are centrioles, the yellow things. Those long lines are the spindle fibers, and what they do is they attach to the center of the chromosomes right in the middle of that X, and that little middle of the X thing is called a centromere. The centromere connects two sister chromatids. So each of those X's is actually two copies of the genetic material. The left half and the right half are identical. They're the exact same. So once everything splits later on, the left half, or the left cell I should say, will get the exact same amount of DNA as the right half. Okay? And again, those are called chromatids. If you look at the real life diagram that's at the bottom there, you can kind of see it's not as pretty as the picture at the top. The real life diagram, things are lined up in the middle there, but again, it's just not as pretty as the little fake picture. It's just a little reminder to everybody. Real life is not as pretty as what you see in your textbook, so that's why we actually have you guys do labs. All right, after we get out of metaphase, we'll move into anaphase. Anaphase, and ana means apart. Anaphase is where we actually take those sister chromatids and we break them apart. So you guys can kind of see the cell is starting to pinch off here as well. The cell membranes pinch towards the center of the cell, and we pull sister chromatids to each side of the cell. The left side is going to get one copy of everything, and the right side is going to get one copy of everything. That way later on when we divide, everything's identical. So you have a gene that helps you do whatever on each side of the cell. Sorry, I couldn't think of an example there. So the real life example, which is on the bottom left or bottom right hand side, you guys can see everything is kind of splitting off to a right side and a left side. Okay, so again, that's anaphase. With telophase, telophase is where we actually start to break down the cell into two new cells. Those sister chromatids have gone to opposite sides of the cell, and now a new nuclear membrane begins to form around them. The cell membrane, again, is pinching off at this point, and this is actually where we begin cytokinesis. So if you guys look at the picture of the cell on top, this is really just showing the nuclear material. If this was a picture that kind of included all the cytokinesis stuff, we would see that we're having like our mitochondria are moving to each side of the cell. So one side's going to get a certain amount of mitochondria, the other side of the cell is going to get a certain amount of mitochondria. We're moving our Golgis and all of our vesicles and peroxisomes and lysosomes and vacuoles, and everything is moving from one side of the cell to the other so that one side has, you know, a couple and the other side has a couple. That way everything is going to be equal. We're going to have two completely identical cells. Everything's going to be equal. The real life picture that's at the bottom of this slide, you guys can kind of see is, you can almost see those little spindle fibers kind of in the middle there. They almost make that little line. But everything has kind of moved back to opposite sides of the cell. So now the cell hasn't completely split yet, you guys can see. But we have two nuclei that are very visible. They're forming there on the left and the right side of that cell. That kind of line that's forming down the middle there, that's something that happens in plant cells. And if you guys have noticed, all of the real life examples that I've showed you are plant cells. That is, the name's escaping me, it's some kind of a plate that's going to turn into a cell wall later on. So if you really want to know, come up and ask me in class, because I'm sure I'm going to look it up right after I'm done making this movie. All right, and then now we've got a couple practice diagrams that I wanted you guys to take a look at. This one, it has them in order. Okay, so prophase, metaphase, anaphase, and telophase. See if you can match them up. I'm going to give you about 15 seconds, and then I'll mark them out. My dogs are going to bark here in a second too, because my wife's coming home, so sorry if you hear all that barking. There you go. All right, if you have tried your answers here, prophase is actually B, metaphase is A. You can kind of see them lining up there in the middle. Remember the M's, middle, meta, they're lining up in the middle. Anaphase, and ana again means going apart, so anaphase is actually D. And then telophase is C. That's where everything's kind of breaking up. All right, so now on this next slide, we're going to actually take a look at all of the different ones, or different phases of mitosis again, except we're going to break them up this time. I didn't put them in order, so see if you can match them up again. All right, a couple seconds, I'll let you guys check these out. All right, telophase is D, prophase is B, anaphase, again, ana means apart, is C, and then metaphase is A, where they line up in the middle. Remember, meta, okay, meta, M, middle, there we go. And last one, there's some real life pictures. You guys have A, B, C, and D on this one. See if you can match up which one is which. This time I didn't put all of them in opposite orders for the wording, so I'm going to give you about 15 seconds to figure this one out, and then we'll see how you do. Again, real life is a little messier than the laboratory and all those diagrams that we see. So, all right, if you've had a couple seconds to check these out, prophase is actually D. You can kind of see it's all in the middle of that cell right there in D. Everything is kind of condensed, so you can see it's not just like a big glob like some of the other cells are just like red blobs in the middle. Metaphase is probably the hardest one here. Metaphase is actually A. Those things are kind of in the middle of that cell. Anaphase is letter B, where things are kind of starting to come apart. And then telophase. This one is in cell C. You can kind of see there's almost got that little white line coming in through the middle there. This one is actually probably already gone all the way through telophase, and it's starting to form that nuclear membrane, and all that DNA is starting to decondense. So if you compare those letter C that has 2 cells there that have broken up, and then letter D, letter C, you can kind of see there the DNA has kind of started to decondense, and there's a lot more red in there than in letter D. So I hope you guys learned something today, and yeah, we'll see you later.